Hello. Very good afternoon to the student who have joined. Can you please let me know in the chat box if my voice is audible to you? Also, please let me know if you can see the presentation. Kindly verify if my voice is audible and also if you can see the presentation correctly. Okay, thank you for confirming. We will start at 4 p.m. Till then, please wait until other students join. Okay, so I think I will wait for a minute, then I will start with the introductions. Okay, so let us start with the introduction of the class. Okay. Yes. So, a very good afternoon to the students who have joined. My name is Mousumi Deb. I am the PMRF Fellow from the Department of Chemistry and Chemical Biology, IIT ISM Dhanbad. I am your TA for this NPTEL course of Medicinal Chemistry, and today it is our week 10 live session. Before starting the session, I would like to point out at a few things that there will be a problem solving session every week, and all these sessions will be on Friday from 4 to 6 p.m. this particular session, and the Google Meet link for the session is same for every week. That is the link which is given here. Okay, and the link is same for our complete semester for the Friday classes which are scheduled from 4 to 6 p.m. and these sessions are live on Google Meet platform. So the important point is like if there is any network disruption from my side, do not leave the session immediately. I will try to rejoin as soon as possible. So please wait, do not leave the classroom. And during the session, the students can post their queries and doubts through the chat box. I am recording the session. That is why I have muted the microphones and cameras of the students. But the chat box is always open. I am monitoring the chat box every now and then. So if you have any doubt, any query or anything, you can let me know through the chat box. Since I am recording it, you can rejoin at any time if there is any network issue from your side. Okay. And I have muted your microphone and camera so that I can record it seamlessly. But the chat box, as I have already said, that it is always open and I'm monitoring the chat box every now and then. So if you have any doubt, any query, anything, you can just let me know through the chat box. Okay. After the class, if any doubt persists, you can drop an email to me. I will share my email ID at the end of the session and I will answer your queries through the email also. Also, there is a discussion forum for this for every the course of the NPTEL, and for this particular medicinal chemistry course also there is a discussion forum. So if you have any doubt, any query, anything, you can let us know in the discussion forum also. We will try to respond to your queries in there also. And the presentation which I am presenting today, as well as the class recording, I will be sharing with the students after the class. Okay, so where you have to find all these recordings, I have already mentioned it couple of times, but I will just show you one more time that there in the medicinal chemistry you have to go to the SWAM portal. When there is the medicinal chemistry announcement section, there will be on the left panel you will find there is a thing called problem solving session January 2025. If you click on that particular thing, you will go to the problem solving session recording. Okay, and after clicking on this link you will be redirected to a page where you can find all the recordings and all the class presentations of the course of the problem solving sessions. Okay, there will be the all the videos and all the materials will be date wise, so you can go through all this in case you miss anything in today's class or any previous class. Okay, so today I have already mentioned it that today it is our week 10 live session, right? So what do we do in every class? We take up already around 20 question of the particular week's course content, and we solve in our class. All the questions which I show you are the multiple choice question, and there will be four options of every question. Okay, after I show you one particular question, there will be I will wait for some time. You have to let me know that A, B, C, or D, which of the following given answers will be correct for that particular question. Okay, and after that I will just check your answers and I will show you the correct answer, and we will discuss that particular topic in detail. So as I have already mentioned, today it is our week 10 live session, right? So we in the week 10 live session, what we are going to do is we are going to discuss the week 10 course content. So what are in the week 10 course content? Let us first see that things. So in the week 10 we have the combinatorial and parallel synthesis. Then we have already actually discussed about little bit about the combinatorial synthesis in the I think seventh or eighth class. I am not particularly correctly remembering, but it was there, the combinatorial chemistry a little bit, which we have all discussed already. So today we will discuss it in detail, the combinatorial and parallel synthesis. Then we will discuss about a little bit about the computer in medicinal chemistry, how computers can be utilized in case of medicinal chemistry. Then we will go to the antibacterial agents. Okay, so antibacterial agents are very important class of therapeutics, and in our course also there are different therapeutics which we will be going to discuss in our coming weeks, like antibacterial, antiviral, anti-cancer, and so on. So antibacterial is the today's main topic, which is the antibacterial agents, and then we have the tutorial on all these topics like the combinatorial, parallel synthesis, computers in medchem, and antibacterial agents. Okay. So let us start with today's first question. Okay, I hope my voice is audible to you and you can see the presentation. If there is any issue regarding anything, please let me know in the chat box. Okay, so now let me just start the today's session. Let me show you the first question of today's session. So the problem number 1, it says that which of the following statements regarding molecular mechanics is false? Your options are: number A, in molecular mechanics, the molecule is treated as a series of spheres connected by springs. Number B, torsional energies are associated with atoms separated from each other by 3 bonds. Number C, molecular mechanics is used to calculate the electronic properties of the molecule. And number D, molecular mechanics follow the classical laws of physics. Which of the following statements is false regarding the molecular mechanics? Read all the options very carefully, and since this is a true false type of question, again you have to be a little bit careful, because many students make mistake in this kind of question. So you have to read all the options very carefully, think, and then let me know in the chat box A, B, C or D which of the following you think is the correct answer for problem number 1. I will wait for 2 minutes, then I will check the chat box, and after that we will discuss it. Okay. Find out which of the following statements regarding molecular mechanics is false. Number A, in molecular mechanics, the molecule is treated as a series of spheres connected by springs. Number B, torsional energies are associated with atoms separated from each other by 3 bonds. Molecular mechanics is used to calculate the electronic properties of the molecule, or molecular mechanics follow the classical laws of physics. Which of the following statements is false regarding molecular mechanics? Let me now check the chat box. Okay, so I have I think 2 answers. One is saying it is number A, one is saying it is number C. Okay, let me check A or C, which of the following will be the correct answer, means the statement which is false. So the correct answer is number C, which says that molecular mechanics is used to calculate electronic properties of the molecule. This statement regarding molecular mechanics is false. Okay, so what are the computational methods that are used to calculate the structure and property data can be split into 2 categories. One is the molecular mechanics and the second one is the quantum mechanics. I know and I think most of you have heard of these 2 terms and especially the quantum mechanics a lot. So molecular, there are 2 categories of this computational methods which are commonly used: molecular mechanics and quantum mechanics. So what is molecular mechanics? So basically in molecular mechanics, equations are used which follows the rules of the classical physics and apply them to nuclei without consideration of the electrons. Okay, this is a very important, that the equations are they follow the rules of classical physics and apply them to the nuclei without consideration of the electrons. In essence, the molecule is treated as a series of spheres, the atoms, okay, and connected by springs, the bond. Okay, so this is all the molecules are treated as a ball, all the spheres, and the springs denotes the bonds. Equations derived from classical mechanics are used to calculate the different interaction and energies, force field resulting from the bond stretching, angle bending, non-bonded interactions, and torsional energies. The torsional energies are associated with the atoms that are separated from each other by 3 bonds. The relative orientation of these atoms are defined by the dihedral or torsional angles. Okay. So equations derived from classical mechanics are used to calculate the different interactions, energies, force field resulting from the bond stretching, angle bending, non-bonded interaction, and torsional energies. Okay. So molecular mechanics is used to calculate electronic properties of the molecule. This statement is false. Okay, it cannot be used to calculate the electronic properties of the molecule. Okay, so now I will go to the next problem. Problem number 2 says, which of the following operations or calculations can't be done using the quantum mechanics? Which of the following operations or calculation cannot be done by using quantum mechanics? Your options are: number A, dipole moment calculation; number B, transition state geometries and energies; number C, energy calculation for specific conformations; and number D, bond dissociation energies. Which of the following operation or calculations cannot be done using quantum mechanics? Dipole moment calculation, transition state, geometries and energies, energy calculations for specific conformations, or bond dissociation energies. Let me know in the chat box A, B, C or D which of the following you think is the correct answer for problem number 2. Which of the following operations or calculations cannot be done using the quantum mechanics? Dipole moment calculation, transition state geometries or energies, energy equations, calculations for specific conformations, and bond dissociation energies. Which of the following operations or calculation cannot be done using quantum mechanics? Okay, let me check the chat box now. Okay, I have received 2 options. One is saying it is number D and another is saying number C. Okay, let me check C or D. Which of the following will be the correct answer or anything else if it is the correct answer, or let me just check. So the correct answer is, it is number C, energy calculations for specific conformations. Okay, this operation or calculation cannot be done by quantum mechanics. So first Molecular mechanics, I have already discussed it in the last slide also. So the choice of method. So what is the choice of method for different operations? So molecular mechanics, okay, is useful for the following operations or calculations, like energy minimization, identifying stable conformation, energy calculations for specific conformations, generating different conformations, and studying the molecular motion. All these things can be studied using the molecular mechanics. And quantum mechanical methods are suitable for calculating the following: molecular orbital energies and coefficients, heat of formation of specific conformation, partial atomic charges calculated from molecular orbital coefficients, electrostatics potential, dipole moment, transition state geometries and energies, and bond dissociation energies. Okay. So these are the methods which can be used for or which can be used for different works. Okay. Again, I will repeat: quantum mechanics are suitable for calculating molecular orbital energies and coefficients, heat of formation for specific conformation, partial atomic charges calculated from molecular orbital coefficients, electrostatic potential, dipole moment, transition state geometries and energies, and bond dissociation energies. And molecular mechanics is useful for the following operations or calculations: energy minimization, identifying stable conformations, energy calculations for specific conformation, generating different conformation, and studying molecular motion. Okay. So energy calculation specific conformation, it cannot be calculated using quantum mechanics, it has to be calculated using molecular mechanics. Okay. So number C, it was the correct answer. Okay. And I think there was a C in the chat box also. So very good. And even if your answer is wrong, it does not matter, since I am not going to mark regarding any answer during the class. But the assignment, you have marks in the assignment. So if you if you pay attention to the class, it will be helpful to you because for your assignment portion also, right? So now I will go to the next problem. Problem number 3 says, which of the following statements regarding solid state synthesis is false? Your options are: number A cross-linked insoluble polymeric support is needed. Number B, an anchor or link should be or anchor or link should be attached with the support. It will be linker. Okay. The anchor should contain reactive functional group. Number D, the protective group of the functional group should involve in the synthetic routes. Okay. Again, I will repeat: which of the following statements regarding solid state synthesis is false? Number A cross-linked insoluble polymeric support is needed. Number B, an anchor or linker should be attached with the support. Number C, the anchor should contain reactive functional group. Number D, the protecting group of the functional group should involve in the synthetic routes. Which of the following statements regarding the solid state synthesis is false? Read all the options carefully, think, and then let me know in the chat box A, B, C, or D which of the following you think is a false statement regarding the solid state synthesis. Which of the following statements regarding solid state synthesis is false? Let me check the chat box. Okay, there is no answer. I hope I am audible, right? Okay, I have got one answer which is saying it is number D. Okay, let me check if number D is the correct answer or not. See, even if your answer is wrong, it's completely fine. Okay, let me check. So the correct answer will be very good. It is number D. The statement is number D statement is false, which says that the protecting group of the functional group should involve in the synthetic routes. This statement is false. Okay. So, what are the essential requirements of the solid phase synthesis? So, the essential requirements are a cross-linked insoluble polymeric support which is inert to the synthetic conditions. For example, a resin bead. An anchor or linker covalently linked to the resin. The anchor has a reacting functional group that can be used to attach the substrate. A bond linking the substrate to the linker, which will be stable to the reaction condition used in the synthesis. A means of cleaving the product or the intermediates from the linker, protecting groups for functional groups not involved in the synthetic route. Okay, so all these are the requirements of the solid phase synthesis mainly. Okay, so the protecting groups of functional group should not be involved in the synthetic route. Okay, this is one of the very important requirements for the solid phase synthesis. So statement number D, which says that the protecting group of the functional group should involve in the synthetic route, it is incorrect. Okay, so very good. The correct answer was number D. So one thing, just it will be a linker. Just correct it. Yeah. So now I will go to the next problem. So problem number 4, it says that Merrifield resin is dash. Your now options are number A, chloroformylated polystyrene, number B, chloroacylated polystyrene, number C, silicon dioxide, which is a polymer of the silicon dioxide, and number D, polymethyl methacrylate. Merrifield resin is, which of the following is the correct answer? What is Merrifield resin? Which of the following will be the correct answer regarding Merrifield resin? Number A, chloroformylated polystyrene. Number B, chloroacylated polystyrene. Number C, silicon dioxide, the polymer of silicon dioxide. And number D, polymethyl methacrylate. Think and then let me know in the chat box A, B, C or D which of the following you think is the correct answer for problem number 4. Merrifield resin is, which of the following is the correct answer? Number A, chloroformylated polystyrene. Number B, chloroacylated polystyrene. Number C, silicon dioxide, the polymer of silicon dioxide. And number D, polymethyl methacrylate. If you have any doubt, any query regarding any question, please let me know in the chat box. Okay. If there is any issue with the voice or presentation also, you can also you can just let me know in the chat box. Let me check the chat box now. Okay. So I have two answers. One is saying it is number D and another one is saying it is number A. Let me check which of the following will be the correct answer, A or D. So, Merrifield resin is, it is number A, chloroformylated polystyrene. Okay. So this is the structure. the chloroformylated polystyrene, which is the Medifil resin. So the first successful example of the solid phase synthesis was the Medifil resin peptide synthesis. Okay, the resin involved consisted of polystyrene beads where the styrene is partially cross-linked with 1% of divinyl benzene. The beads are derivatized with a chloromethyl group, the anchor or linker to which the amino acids can be coupled via the ester group. The ester group is stable to the reaction condition used in peptide synthesis but can be clipped at the end of the synthesis using the vigorous acidic conditions like hydrofluoric acid. So this is the peptide synthesis on a solid support. Okay. So here first one is our resin bit, as I have already mentioned it here. This is our Medifil resin, which has this chloromethyl group here, and it does in this position. And then we have amino acid. For the particular amino acid, as you can see, the nitrogen, the NH2 is blocked. Okay. And what is the protecting group? This is a Bock protecting group. The full form of Bock is a butyl oxycarbonyl, okay, or TBOCO. So this is the N protected amino acid. So the first step is the formation of the ester. Okay, as you can see, there is a sorry, no, not the yes, the first step is the formation of this ester. So as you can see, there is a normal reaction. Okay, and then what happens? The reprotection with the TFA. The TFA is the trifluoroacetic acid. So after attachment of the first amino acid, the trifluoroacetic acid is applied, so that which causes the Bock deprotection. Okay, so once the Bock group leaves, we have the NH2 is now free. Then another N protected amino acid comes here, and the coupling happens, amide coupling happens, and this thing repeats for the couple of times till we get the peptide chain. And after that, at the end, what happens? This particular ester group, it can be clipped, okay, and we will get a complete peptide by use of the vigorous acidic workup using using strong acid like the hydrofluoric acid. Okay, so then we will get the peptide chain and we will get our resin bit back. So this is the how the Medifil resin, how the solid phase system peptide synthesis happens on Medifil resin. This is a very good example. The steps are very simple and also I mean easy, but you have to practice it for a couple of times so to get it right completely. Okay. So the correct answer was number A, chloro with formylated polystyrene is the Medifil resin, which has this structure. Okay. Now I will go to the next problem. So problem number 5 says, which of the following statements is incorrect regarding the microwave technology? Your options are: number A, it requires shorter reaction time. Number B, coupling reactions cannot be performed using this. Number C, it leads to the fewer side reactions. And number D, it is employed in library synthesis. Which of the following statements is incorrect regarding the microwave technology? Number A, it requires shorter reaction time. Number B, coupling reactions cannot be performed using this. Number C, it leads to fewer side reactions. Number D, it is employed in library synthesis. Which of the following statements is incorrect regarding microwave technology? Again, think, read the options carefully, and then let me know A, B, C, or D, which of the following you think is the correct answer for problem number 5. So, which of the following statements is incorrect regarding the microwave technology? Let me check the chat box. Okay, I have received two answers. Both are saying it is the number B, which is the correct answer. Let me check number B if it is the correct answer or not. So, the statement which is incorrect regarding the microwave technology, it is very good. It is number B. Coupling reactions cannot be performed using this. This statement is false. So basically, what is the microwave technology? So drug discovery is a very expensive process, and microwave assisted organic synthesis, or MAOS, is proving to be the very useful tool for the accelerating synthesis and making the process more efficient. There are many examples of thermal reactions that take several hours to complete using the heaters or oil baths, but which are carried out in minutes using the microwave conditions. There is a much greater efficiency of energy transfer using microwave technology, which accounts for the faster reaction times. Moreover, yields can sometimes be improved dramatically, with less decomposition and fewer side reactions. Specially designed microwave units are now commonly employed in library synthesis. Examples of the reactions that have been carried out using microwave technology include the formation of amides from acids and amines without the need of the coupling reaction, metal catalyzed Suzuki couplings, which can be performed even on usually unreactive aryl chlorides, the metal mediated reduction aminations, and the reductions and many reduction which took 24 hours using conventional heating, only 15 minutes using the microwave heating. Okay. So these are the example of the microwave assisted reactions which can be done. So amide coupling is one of the those reactions which are sometimes very difficult to especially using only acid and amine. It is very difficult. So we actually need the reactive part of the acid like acyl chloride, and also sometimes we note the coupling reagents like DCC and so on, or HBTU, and all the amide coupling reagents. There is a many amide coupling reagents which are required, and also sometimes high temperatures are required because due to the amide coupling. But using the microwave technology, it can be done very easily in a very shorter reaction time. The Suzuki coupling reaction is an example of Suzuki coupling reaction, which also can be done using the particular microwave synthesis technology in very shorter reaction time. Also, we have other examples like reduction. For example, here the reduction can happen. The amination can also happen. So all these are the reactions which can be done in the microwave technology. In the medicinal chemistry, sometimes we actually need a very efficient reaction systems or the reaction conditions so that we can form a large number of molecules in a shorter reaction time. So in those cases, microwave technology proves to be a very efficient tool in case of the medicinal chemistry synthesis. Okay. Now let's go to the next problem. Problem number 6 says, which of the following reactions is used in the dynamic combinatorial synthesis of vancomycin dimers? Your options are number A, Suzuki coupling, number B, Boltzmann cyclization, number C, olefin metathesis, and number D, Michael reaction. Which of the following reactions is used in the dynamic combinatorial synthesis of vancomycin dimers? Which of the following reactions are used in is used in dynamic combinatorial synthesis of vancomycin dimers? Suzuki coupling, Boltzmann cyclization, olefin metathesis, or Michael reaction? A, B, C, or D? Let me know in the chat box which of the following you think is the correct answer. I will give you I will give you 2 minutes. Then I will check the chat box. Okay. Which of the following reactions used in the dynamic combinatorial synthesis of vancomycin dimers? Suzuki coupling reaction, Boltzmann cyclization reactions, olefin metathesis, or Michael reaction? Let me check. Okay, I have got one answer. It's saying that is the number B. Let me check if number B is the correct answer or not. So, the reaction which is used in the dynamic combinatorial synthesis of the vancomycin dimers is, it is number C, olefin metathesis. Okay, so now let us just check. So the dynamic combinatorial synthesis of the vancomycin dimer. So vancomycin is an antibiotic that works because it marks and the building blocks required for the bacterial cell wall synthesis. Okay, binding takes place specifically between the antibiotic and the peptide sequence L-lysine D-ALA, which is present in the building block. It is also known that the binding promotes the dimerization of the vancomycin target complex, which suggests that covalently linked vancomycin dimers might be more effective antibacterial agents than vancomycin itself. A dynamic combinatorial synthesis was carried out to synthesize a variety of different vancomycin dimer covalently linked by the bridges of different lengths. The vancomycin monomers used had been modified such that they contain the long chain alkyl substituents with the double bonds at the end. The reaction between the double bonds and the presence catalyst then led to the bridge formation through the reaction known as the olefin metathesis. Okay, so first let us see what is the olefin metathesis. So basically olefin metathesis, when we have a terminal double bond in both cases, this is basically joining the particular two double bonds here like this, and here we get the particular olefin which has the cis trans isomers. Okay, and this is the so the tripeptide target was present to accelerate the rate of the bridge formation, which to promote the formation of the vancomycin dimers having the ideal bridge length. So this is the figure. This is the tripeptide, this is the vancomycin monomer, and this is the ligand binding to the vancomycin monomer, and then so as you can see there is a double bond here. So this is at the terminal position. Okay, and the vancomycin monomers bridge the tripeptide, which encourages the self assembly of non-covalently linked dimers. Once formed, those dimers having the correct length of the substituents are more likely to react together to form the covalent bridge. Having established the optimum length of the bridge, another experiment was carried out on 8 vancomycin monomers which had the correct length of the tether and varied slightly in their structure. The mixture of 36 possible products was analyzed by the mass spectrometry to indicate the relative proportion of the each dimer formed. 11 of the 36 compounds were then synthesized separately, and it was found that the antibacterial activity matched the level of the amplification, that is, the compounds present in the greater quantities had the greater reactivity. Okay. So this is only for your understanding. Okay, you don't need to memorize all these things. Read and understand all those things, and remember that how this olefin metathesis happens in the combinatorial synthesis of the vancomycin dimer. Okay, so this is the whole process, and this is the basic olefin metathesis reaction. Okay, now I will go to the next problem. Problem number 7 says, which of the following is the privileged scaffold for the medicinal chemistry? Your options are number A, benzodiazepine, number B, hydantoin, number C, tetrahydroisoquinoline, number D, all of the above. So, which of the following is or are the privileged scaffold for the medicinal chemistry? Benzodiazepine, hydantoin, tetrahydroisoquinoline, or all of the above? Let me know in the chat box A, B, C or D, which of the following you think is the correct answer? Which of the following you think is the privileged scaffold in the medicinal chemistry? Which of the following is the privileged scaffold in the medicinal chemistry? Benzodiazepine, hydantoin, tetrahydroisoquinone, or all of the above? I will wait for 2 minutes, then I will check the chat box, and after that I will show you the correct answer and we will discuss about it. So which of the following is the privileged scaffold for the medicinal chemistry? Benzodiazepine, hydantoin, tetrahydroisoquinone, or all of the above? Let me check. What are your answers? Okay. So I think I have got two answers. One is saying it is the number D, another one is saying it is the number B. Okay, let me check which of the following is the correct answer. So the privileged scaffold in for the medicinal chemistry is, it is number D, all of the above. So benzodiazepine, hydantoin, tetrahydroisoquinone, all of these are the privileged scaffolds for the medicinal chemistry, and there are also a few couple of more scaffolds. So basically, most scaffolds are constructed using the synthetic route employed for the solid phase synthesis, and this also determines the number and the variety of substituent that can be attached to the scaffold. The ideal scaffold should be smaller should be small in order to allow wide variation of the substituent. It should also have the substituent widely dispersed around the structure spider-like rather than the restricted in the one part of structure tadpole-like. If the conformation space around is fully exposed. Finally, the synthesis should allow each of the substituent to be varied independently to each other. Scaffolds can be flexible, for example, peptide backbone, or rigid like cyclic system. They may contain heteroatoms and capable of forming useful bonding interactions with the binding site, or they may not. Some scaffolds are already common in medicinal chemistry like benzodiazepine, hydantoin, tetrahydroisoquinone, benzoyl biaryls, and are associated with a diverse range of activities. Such scaffolds are termed as the privileged scaffold. So this is the dipeptide, this is the glucose, this is the steroid, then hydantoin, this is the 1,4-benzodiazepine. Okay, this is the pyridine structure, this is the labitalactam structure, it is the indole structure, then we have biphenyl pyrimidine and diphenyl methane structure. So all the structure in the blue is the known as the privileged scaffold, and these all the things are the functionalization. So benzodiazepines, hydantoins, beta-lactams, and pyrimidines are examples of extremely good scaffolds. They all have the small molecular weights, and there are various synthetic routes available which produce the substituent patterns required to fully explore the conformation space about them. For example, it is possible to synthesize the benzodiazepines such that there are variable substituents around the whole structure. Peptide scaffolds are flexible scaffolds which have the capacity to form the hydrogen bonds with the target binding sites. They are easy to synthesize with a large variety of different substituents are possible by using amino acid building blocks. Further substitution is possible on the terminal amino and carboxylic acid function. The substituents are widely distributed along the peptide chain, allowing a good exploration of conformation space. If we consider Lipinski's rule of 5, the peptide scaffold should ideally be restricted to di or tripeptide in order to keep the molecular weight below 500. It is interesting to note that orally active antihypertensive agents like captopril and enalapril, the dipeptide, like where the large peptides such as the enkephalins are not orally active. Oral activity has been a problem with the HIV protease inhibitors that have molecular weight over 500. Okay. Some of the scaffolds as shown in the diagram have variable disadvantages. For example, although glucose has a small molecular weight, it's possibly of 5 variable substituent. It can cause multiple hydroxyl group attaching different substituent to the similar groups usually requires complex deprotection strategy. Nevertheless, the potential of sugar-based drugs is also great, and a lot of progress has been made developing the solid phase synthesis based on the sugar scaffolds. Steroids might be attractive, might also appear attractive as a scaffolds. However, the molecular weight of steroid skeleton itself limits the size of the number of substituents can be added if you wish to keep the overall molecular weight below 500. The indole scaffold suffers a disadvantage of having various substituents located on the same region of the molecule, preventing the full exploration of the conformation space. That is the tadpole-like structure. Okay, so these are the various examples of the scaffolds, and the previous scaffolds, as I have already mentioned, are there are several. Okay, so all of the above was the correct answer. Now I will go to the next problem. So problem number 8 says, which of the following statements is not true for the bacteriostatic agents? Your options are: number A, the agents stop the growth of the bacteria; number B, the bacterial population remains the same; number C, the agents destroy the bacteria; number D, the agents inhibit the processes like the replication, translation and the transcription. Which of the following statements is not true for the bacteriostatic agents? Number A, they stop the growth of the bacteria. Number B, the bacterial population remains the same. Number C, the agents destroy the bacteria. Number D, the agents inhibit the processes like replication, translation and transcription. Let me know, A, B, C or D, which of the following you think is the statement which is not true regarding the bacteriostatic agents. So let me check. Okay, so I have one answer which is saying it is the number C. Let me check if number C is the correct answer or not. Yes, number C is the correct answer, which says that the agents destroy the bacteria. So now let us discuss. So the definition: the bacteriostatic antimicrobial agents that reversibly inhibit the growth of the bacteria are called the bacteriostatic. For example, the tetracyclines, chloramphenicol, bactericidal. Those with an irreversible lethal action on the bacteria are known as the bactericidal, like penicillin, isoniazid. So these are the differences between the bactericidal and bacteriostatic. So bactericidal refers to the agents that kill the bacteria, and bacteriostatic refers to the antibody that prevent the growth of the bacteria. So the action of the bactericidal is irreversible, whereas the action of the bacteriostatic is reversible. Inhibit the cell wall. The bactericidal agents inhibit the cell walls formation of the bacteria. Bacteriostatic agents inhibit the DNA replication and the protein synthesis of the bacteria. Bactericidal agents do not work with the immune system of the host, whereas the bacteriostatic agents work with the immune system of the host to prevent the growth and the reproduction of the bacteria. Bactericidal agents, the MBC refers to the concentration of the drug required to kill the 99.99% of the bacterial population. MIC, or minimum inhibitory drug concentration, which inhibits the bacterial growth. Example of bactericidal include the beta-lactam antibiotics, cephalosporin, vancomycin. The example of bacteriostatics is the tetracycline, bacteriomycin, chloramphenicol, and sulfonamides. Okay. So these are the differences and the comparison between the bactericidal and bacteriostatic agents. So statement number C, which says that these agents destroy the bacteria, is not true. These agents inhibit the different cellular activity and different cellular processes, and it stops the growth of the bacteria. They cannot kill the bacteria directly. Okay. So statement number C, yes, it was the correct answer. Now problem number 9, it says that which of the following is not a semi-synthetic antibacterial agent? Your options are number A, ampicillin, number B, carbonicillin, number C, cephalexin, number D, sulfonamide. Which of the following is not a semi-synthetic antibacterial agent? Ampicillin, carbonicillin, cephalexin, or sulfonamide? Let me know in the chat box, A, B, C, or D, which of the following you think is the correct answer, or which of the following is not a semi-synthetic antibacterial agent? Ampicillin, carbonicillin, cephalexin, or sulfonamide? Which of the following is not a semi-synthetic antibacterial agent? Let me check the chat box. Okay, number D. Let me check if number D is the correct answer or not. So, the following, which is not a semi-synthetic antibacterial agent, will be number D. Very good. It is the sulfonamide. So sources of antibacterial agents. So the sources of the antibacterial agents can be several, like the natural sources, mainly the fungal sources. The original antibiotics were derived from the fungal sources, for example, and this fungal sources can be termed as the natural antibiotics. For example, the benzyl penicillin and gentamicin are the natural antibiotic. We have the semi-synthetic ones, the chemically altered natural compound developed to decrease the toxicity and to increase the effectiveness. For example, ampicillin and amikacin and the semi-synthetic antibiotics. Synthetic chemically designed in the lab, bacteria are not exposed to the compounds until they are released. They are also designed to have much greater effectiveness and less toxicity. For example, moxifloxacin and norfloxacin are the synthetic antibiotics. So, sulfonamide is a synthetic antibacterial agent. This is the structure of the sulfonamide. Okay, so what are the sulfonamides or sulfa drugs? So sulfonamides are the antibiotics that have structural similarities with the para-aminobenzoic acid containing the sulfonamide SO2NH2 groups in their chemical structure. They have bacteriostatic action on the gram-positive and gram-negative bacteria. Sulfadiazine, methoxazole, cotrimazole, trimethoprim. These all are the example of the sulfonamide or sulfa drugs. What are the mechanism of action? The sulfonamides compete with the para-aminobenzoic acid, which is an essential precursor in the synthesis of the folic acid required for the synthesis of DNA and RNA in bacteria. They inhibit the bacterial folate synthesis, lead to the impairment of the DNA formation. So this is the mechanism of action of the sulfonamides or sulfa drug. Okay. So basically, I have already mentioned the sulfonamide. They have the structure similarly to the para-aminobenzoic acid. Okay, and this para-aminobenzoic acid is an essential precursor for the synthesis of folic acid. And so, as you can see, this is the how sulfonamide prevents the para-aminobenzoic acid from binding to the binding by mimicking mimicking this. So this is the para-aminobenzoic acid in the active site. It interacts with the hydrogen bond with this NH2 portion, favorable interaction with the ring, and ionic bond with the COO minus group. Okay, and here also the sulfonamide, it mimics the para-aminobenzoic acid, binds to the active site, and in this way it inhibit this PABA from binding to the active site, and in this way it hinders the folate synthesis. And the sulfonamides are very good example of the reversible inhibitors. Okay. It acts as a reversible inhibitors where it and the competitive inhibitors where it competes with the substrate for the active site. Okay, it mimics the substrate and binds to the active site. Okay. So the correct answer was number D, which says the sulfonamide is not a semi-synthetic antibacterial agent. It is a completely synthetic antibacterial agent. Okay. So now I will go to the next problem. This was problem number 9. Now let us go to the next problem. So problem number 10, it says that which of the following antibacterial agents works by affecting the bacterial plasma membrane permeability? Your options are number A, polymyxin, number B, penicillin, number C, aminoglycoside, number D, nalidixic acid. Which of the following antibacterial agents works by affecting the bacterial plasma membrane permeability? Your options are polymyxin, penicillin, aminoglycoside, or nalidixic acid. Let me know in the chat box A, B, C or D which of the following you think is the correct answer from the problem number 10. Which of the following antibacterial agents works by affecting the bacterial plasma membrane permeability? Polymyxin, penicillin, aminoglycoside or nalidixic acid? Even if your answer is wrong, it won't matter since I am not going to give you any negative marking. But please try to answer the questions. In the chat box, let me know A, B, C or D. Any one option which you think is the correct answer. Which of the following antibacterial agents works by affecting the bacterial plasma membrane permeability? Polymyxin, penicillin, aminoglycoside or nalidixic acid? I will wait for 2 to 3 minutes, then I will check the chat box, okay? So which of the following antibacterial agent works by affecting the bacterial plasma membrane permeability? Let me check the answers. Okay, I have got one answer which is saying number B, another one answer which says it is penicillin. Okay, let me check if which of the following answer is correct. So the correct answer is number A, polymyxin. Okay, polymyxin, they works by affecting the bacterial plasma membrane permeability. Okay, the penicillin, it works by inhibiting the cell wall synthesis. So there is a difference between the cell wall and the plasma membrane. I will just mention it shortly. Just let me check the other answers. Okay, so both answers were penicillins. Okay, so basically penicillin works by a different manner. Okay, so the question was asked that which antibacterial agents work by affecting the bacterial plasma membrane permeability. Okay, so the correct answer is number A, polymyxin. So now let me just discuss it. So this is the bacterial cell wall and bacterial cell and different drug targets. Okay, so as you can see, there are rifamycin, sulfonamides, penicillin, cephalosporin, and cycloserine, polymyxin. Then we have chloramphenicol, streptomycin, tetracycline, ribosome. Okay, so what are these things? First, let us understand. Okay, so this is the outer membrane. Okay, and this outer membrane is present. So the outermost membrane is the known as the outer membrane, which is the only for the gram-negative bacteria. After that, we have the cell wall, and in between we have the periplasmic space. This is the cell wall, and after the cell wall, we have this. This is the plasma membrane. Okay, so first there is an outer membrane, then we have the periplasmic membrane, then cell wall, then we have the plasma membrane. These are the ribosomes, and these are the nuclear material like DNA, RNA, and this is the cytoplasm. So as you can see, there are different antibacterial agents, and it is mentioned where they act actually. So quinolones, aminoacridins, rifamycin, all they act on the DNA nuclear material, DNA, that is the DNA RNA. Ribosomes, the chloramphenicol, streptomycin, tetracycline, they actually attacks or they works on the this particular ribosomes. Then polymyxins act on the plasma membrane. This is the plasma membrane. The question box asks regarding the plasma membrane, not the cell wall. Penicillin, cephalosporin, cycloserine, they work on the cell wall. Okay, and sulfonamides, they works on the different enzymes. So these are the different antibacterial agents and their drug targets. So mechanism of antibacterial agents. So the five main mechanisms by which the antibacterial agents can act. Inhibition of the cell metabolism. Antibacterial agents which inhibit the cell metabolism are called the antimetabolites. Okay, these compounds inhibit the, so these are the very important thing. Let me just highlight this thing. So there are five main mechanisms for the by which the antibacterial agents can act. Okay, those we have first one is the inhibition of the cell metabolism. Okay, so antibacterial agents which inhibit the cell metabolism are known as the antimetabolites. Okay, antimetabolites, remember this term, it is very important. We will discuss it in later. These compounds inhibit the metabolism of a microorganism but not the metabolism of the host. They can do it by inhibiting the enzyme catalyzed reaction which is present in the bacterial cell but not in the animal cells. It is best known example of the antibacterial agent acting in this way were the sulfonamides. It is also possible for antibacterial agents to show the selectivity against the enzymes which are present in the both bacterial and mammalian cell as long as there are significant differences in the structure between the two. Then second mechanism is the inhibition of the bacterial cell wall synthesis. So it leads to the bacterial cell lysis and death. Agents operating in this way include the penicillin, cephalosporin, and glycopeptides such as vancomycin. As animal cells do not have a cell wall, they are not affected by these cells. Okay, this is a very important thing. Interaction with the plasma membrane. Some antibacterial agents interact with the plasma membrane of the bacterial cell to affect the membrane permeability. This has a fatal results on the cell. Polymyxin and tyrosine operate this way. Then we have the disruption of the protein synthesis. Means that the essential proteins and enzymes required for the cell survival can no longer be made. Agents which disrupt protein synthesis include the rifamycin, aminoglycoside, tetracyclines, and chloramphenicol. Then we have the inhibition of the nucleic acid transcription and replication. Okay. It prevents the cell division and other or the synthesis of the essential proteins. Agents acting in this way include the nalidixic acid and proflavin. So these are the 5 main mechanisms by which the antibacterial agents can act. So the question was asked that which can affect the bacterial plasma membrane permeability. It is the polymyxin. So this is the structure of the polymyxin. It's a very complicated structure. You do not need to memorize the structure. It is I have just only given it so that you can just understand how the structure looks. Okay. So this was the problem number 10. Now I will go to the next problem. So problem number 11 says, which of the following is not an example of anti-metabolites used as the antibacterial therapeutic? Your options are number A, trimethoprim, number B, doxycycline, number C, sulfone, and number D, sulfamethoxazole. Sulfamethoxazole. Which of the following is not an example of the antimetabolites used as the antibacterial therapeutics? I have already mentioned the antimetabolites which are called antimetabolites in the last slide itself. So now you have to identify among the these 4, trimethoprim, doxycycline, sulfone and sulfamethoxazole, which of the following is not an example of the antimetabolites? Which of the following is not an example of antimetabolites? Let me know in the chat box, A, B, C or D. Which of the following is not an example of antimetabolites used as antibacterial therapeutics? Trimethoprim, doxycycline, sulfone or sulfamethoxazole? Let me know A, B, C or D. Which you think is the correct answer? Let me check the chat box now. Okay. I have one answer which is saying doxycycline. Let me check if it is the correct answer or not. So you are saying that doxycycline is not the antimetabolites, right? Let me check. So the correct answer will be very good. It is number B, doxycycline. So doxycycline is not an example of antimetabolites. Okay. How doxycycline works, we will discuss it in later. So the antimetabolites, these are the examples of the antimetabolites used in the as antibacterial agents. We have the trimethoprim, then we have the sulfones, and we have the sulfamethoxazole. So trimethoprim is an orally active diaminopyrimidine structure. Okay, so this is the pyrimidine, and we have 2 aminos. So this is a diaminopyrimidine structure, which has proved to be highly selective antibacterial and antimalarial agent. It acts against the dihydrofolate reductase, the enzyme which carries out the conversion of the dihydrofolate to tetrahydrofolate, leading to the inhibition of the DNA synthesis and cell growth. Dihydrofolate reductase is present in mammalian cells as well as the bacterial cells, but mutations over millions of years have resulted in a significant difference in the structure between the 2 enzymes, such that trimethoprim recognizes and inhibits the bacterial enzyme more strongly. In fact, trimethoprim is 1 lakh times more active against the bacterial enzyme. Trimethoprim is often given in conjugation with the sulfonamide sulfamethoxazole in a preparation called clotrimazole. Okay, the sulfonamide inhibit the incorporation of the para-aminobenzoic acid into dihydrofolate, while trimethoprim inhibits the dihydrofolate reductase. Therefore, two enzymes in one biosynthetic routes are included. This is very effective method of inhibiting the biosynthetic route that has taken advantage of the doses of both drugs and can be kept down to the safety level. To get the same level of inhibition using single drug, the dose level has to be much higher, leading to the possible side effects. This approach has been described as a sequential blocking. So this is a very interesting term which we have come upon. So just let me mark it for you. So this is another question can generate from this. What is the sequential blocking? And this is a very good example how trimethoprim and sulfonamides are actually I mean they are actually involved in the sequential blocking. Resistance to trimethoprim has been observed in strains of E. coli which produce a new form of the target enzyme that has less affinity to the drug. Sulfones, this one, they are most important drugs used in the treatment of leprosy and believe they inhibit the same bacterial enzyme inhibited by the sulfonamides, the dihydrofolate synthetase. Okay, so yeah, these are the examples of the anti-metabolites which are used in the antibacterial therapeutics. Now I will go to the next problem. Problem number 12 says, mechanism of action of penicillin involves the inhibition of a particular enzymes in the bacterial cell wall. Identify it. Your options are number A, carboxypeptidase, number B, transpeptidase, number C, glycosylase, and number D, phosphatase. Which of the following enzyme is inhibited by the penicillin in the bacterial cell wall? Carboxypeptidase, transpeptidase, glycosylase, or phosphatase. Let me know in the chat box A, B, C, or D which of the following you think is the correct enzyme that is involved in the inhibition of bacterial cell wall in by penicillin. A, B, C, or D. Let me know. I will wait for 2 to 3 minutes. I will wait for 2 minutes. Then I will check the chat box. Okay. Mechanism of action of penicillin involves the inhibition of particular enzyme or bacterial cell wall. Identify the enzyme. Carboxypeptidase, transpeptidase, glycosylase, or phosphatase. Mechanism of action of penicillin involves the inhibition of particular enzyme in bacterial cell wall. Identify it. Let me check the chat box now. I think it is a very easy question and I know all of you can answer it correctly. Okay, I have the answers which are saying it is number B, and one answer I have received which is the saying it is number C. Okay. So the correct answer will be it is number B. The transpeptidase is the enzyme which is involved in the mechanism of action of penicillin. So basically this is the structure of the penicillin. Okay, the structure of penicillin is very important, and I will suggest that you have to remember this thing. So in the penicillin, we have the part which is it's called the 6-amino penicillinic acid or 6-APA, this blue one. And here we have this is the structure where we have this is the thiazidylone. Okay, this is the thiazolidine ring. This one, this 5-member ring. We have a beta-lactam ring. This one, this 4-member ring. Then we have a acyl side chain which has the R functional group. R is the benzyl penicillin. In case the benzyl penicillin, this is the R, and on the phenoxymethyl penicillin or penicillin V, it is the R is the this one. Okay, so this is the structure of penicillin. So, mechanism of action of penicillin. The structure of the cell wall. In order to understand penicillin's mechanism of action, we have to first look at the structure of the bacterial cell wall, okay, and the mechanism by which it is formed. The bacteria have cell walls in order to survive in the large range of environmental conditions such as varying pH, temperature and osmotic pressure. Without a cell wall, our water would continually enter the cell as a result of the osmotic pressure, causing the cell to swell and burst, which is the lysis. The cell wall is very porous and does not block the entry of the water, but it does not prevent the cell swelling. Animal cells do not have the cell wall, making it perfect target for the antibacterial agents such as penicillins. The wall of is the peptidoglycan structure. Okay, in other words, it is made up of the peptide and sugar units. The structure of the cell wall consists the parallel series of the sugar backbone containing two types of sugar, N-acyl muramic acid or NAM and N-acyl glutasamine or NAG. Okay, so this is the structure, the sugar containing the cell wall structure in bacteria. When R equal to H, there is R equal to H, it is the NAG or N-acyl glutasamine, and when the R equal to CH, MECOOH, it is the N-acyl muramic acid or NAM. Okay. The peptide chains are bound to the NAM sugars, and it is interesting to note that the presence of D amino acids in this chain. In human biochemistry, there is only L amino acid, whereas the bacteria have racemase enzyme that convert L amino acids to D amino acid. In the final stage of cell wall synthesis, the peptide chains are linked together by the displacement of D-alanine by one chain of the glycine to another. The 30 enzymes are involved in the overall biosynthesis of the cell wall, but it is the final cross-linking reaction which is inhibited by the penicillin. This thing is very important to note down, that although about 30 enzymes are involved in the overall biosynthesis of the bacterial cell wall, it is the final cross-linking reaction which is inhibited by the penicillin. Okay, this leads to the cell wall framework to not longer interlink. As a result, the wall becomes fragile and no longer prevent the cell from swelling and bursting. The enzyme is responsible for the cross-linking reaction, is known as the transpeptidase reaction. Okay, the transpeptidase enzyme. Sorry, so there are several types of enzyme which when vary in character but bacterial species together, they are all inhibited by various degrees of the penicillin. This is how the penicillin inhibition happens. This is the mechanism of the transpeptidase cross-linking and penicillin inhibition. Okay, so this is the transpeptidase enzyme. As you can see, we have a serine and lysine unit here and here. So we have a serine and lysine here, and this is the how the penicillin it acts. So it comes here and binds with the serine. When the serine OH it attacks to the nucleophilic attack on this position, and then it binds to this with this here, and the lysine is also involved in this interaction, anionic interaction. Once it binds, the polypeptide chain, it cannot come here. It is blocked, and that's how the particular thing is happens. Okay, the cell wall synthesis inhibition happens by penicillin. This is the mechanism of action of the penicillin. So remember these things, that what is the enzyme, how it happens, and the structure of penicillin are very important. Okay, now I will go to the next problem. Problem number 13 says, which of the following is an incorrect statement regarding the SAR of penicillin? SAR means the structure activity relationship of the penicillin. So I have just shown you the structure. This question is related to the structure of the penicillin, which I have shown in the just last slide. Okay. The first one, number A says the beta-lactam ring is essential. Number B, a bicyclic system is essential. Number C, methyl group in the C2 position are essential. Number D, free carboxylic acid group is essential. So which of the following statements is incorrect regarding the SAR of penicillin? Which of the following statements is incorrect regarding the SAR of penicillin? Number A, beta-lactam ring is essential. Number B, a bicyclic system is essential. Number C, methyl groups in C2 position are essential. Number D, free carboxylic acid group is essential. Which will be the correct answer? Which will be the incorrect statement regarding the SAR of penicillin? I will give you 2 minutes, then I will check the chat box, okay? Which of the following is an incorrect statement regarding the SAR of penicillin? Let me check the chat box. Okay, two answers I have received. One is number D and another is saying it is number C. Okay, let me check which is the correct answer. So, the incorrect statement regarding the SAR of penicillin will be number C, which says that methyl group's in situ position is essential. So this is the SAR of penicillin, and this is a very important thing. Okay, this is the structure of the penicillin. As you can see, I have already mentioned there are two bicyclic ring system. One is the lactam ring and is the thiazolidine ring. Okay, so these things are essential. One is the amide. This amide linkage is essential. Then the cis stereochemistry of the hydrogens is essential. This lactam is essential. Okay, and the bicyclic system is essential. Free carboxylic acid is also essential. So these things are essential. So the large number of penicillin analogues have been synthesized and studies. The results of these studies led to the SR of the SR conclusions. Number one is the strain beta-lactam ring is essential. The free carboxylic acid is essential. This is usually ionized or penicillins are administered as sodium and potassium salts. The carboxylate ion binds to the charged nitrogen of the lysine residue in the binding site. The bicyclic system is essentially important. This confers the further strains and the beta-lactam ring, and the greater of the strain and the greater is the activity, but the greater is the instability of the molecule. To the other factors, the acyl amino side chain is essential. The sulfur is usual but not essential. The stereochemistry of the bicyclic ring with respect to the acyl amino side chain is also essential. The results of this analysis led to the inevitable conclusion that a very little variation is tolerated in the penicillin nucleus, and any variation is restricted to the acyl amino side chain. Okay, this acyl amino side chain where the variation can be made, other are the cannot be changed. So the number C, which says the methyl group in situ position, this one is essential. It is false. It is not essential. Okay, other things are essential, which I have already mentioned in the different points. So now this was the problem number 13. I will go to the next problem. Problem number 14 says pivamycin is a prodrug of dash. Fill in the blanks. Your options are number A penicillin, number B cephalosporin, number C ampicillin, and number D amoxicillin. Pivamycin is prodrug of which compound? Penicillin, cephalosporin, ampicillin, or amoxicillin? Pivamycin is a prodrug of which one? Penicillin, cephalosporin, ampicillin, or amoxicillin? I will wait for 2 minutes. Let me know in the chat box which of the following you think is the correct answer. Bivamycin is a prodrug of what? Penicillin, cephalosporin, ampicillin, amoxicillin. Bivamycin is a prodrug of which one? Let me check the chat box. Okay, I have received two answers and both are saying it is the number C, I think. Yeah, both are saying it is number C. Let me check if number C is the correct answer or not. So, pivamycin is the prodrug of, very good, it is number C, ampicillin. So, ampicillin prodrug. So, pivamycin, talamycin, and vancomycin are the prodrugs of the ampicillin. Okay, so these are the structures. This is the ampicillin, and these are the prodrugs of ampicillin, pivamycin, talamycin, and vancomycin. So this is the R which is varies here in all these prodrugs. Okay, so in these three cases, the esters used to mark the carboxylic acid group seem rather elaborate, and one may ask why the simple methyl ester cannot be used. Okay, so this is the simple. As you can see, there is a free carboxylate group. We are already on the bicyclic system, and this is the esters of that carboxylate group. And these all esters are very complicated esters. If you can see the structures, okay, they may seem complicated, and the question may arise that why not simple methyl group can be used as R. Okay, the answer is that methyl esters of penicillins are not metabolized in humans. The bulky penicillin skeleton is so close to the ester that it acts as a steric shield and prevents the esterase enzymes that catalyze the reaction from accepting the penicillin ester as the substrate. Fortunately, acyloxymethyl esters are susceptible to the esterases. These extended esters contain the second ester group further away from the penicillin nucleus. So as you can see, all these things they contain another ester group. Okay, and which is more exposed to attack. The hydrolysis produces the products are inherently unstable and decompose spontaneously to release the formaldehyde and reveal the free carboxylic acid. The release of the formaldehyde is not ideal. It is the toxic chemical. However, it is formed naturally in the body through the enzymatic demethylation of the various compounds found in the diet, and levels produced from the product describes the cause this problem. Moreover, the drugs are only taken in the short period of the time. Such extended esters can be used to prepare the prodrugs of the penicillin, but one has to be careful that one does not go for the other extreme and make the penicillin too lipophilic. For example, one acyloxyl ester of the penicillin G is so lipophilic that it has a poor solubility of water. Fortunately, the problem can be avoided easily by making the extended ester more polar. by attaching vanillin. So this is the different examples. So this is the mechanism by which the acyloxymethyl esters are hydrolyzed, okay, by the esterases by producing the formaldehyde, and this is the example how the polar extended ester of the penicillin G. Okay, so yeah, number C was the correct answer. Now I will go to the next problem. Problem number 15 says erythromycin belongs to dash class of antibiotics. Your options are number A macrolide, number B tetracycline, number C oxalidiolone, number D quinolone. So erythromycin belongs to which class of antibiotics? Macrolide, tetracycline, oxalidiolone or quinolones? Erythromycin belongs to dash class of antibiotics. Macrolide, tetracycline, oxalidiolone, quinolone. I will wait for 2 minutes, then I will check the chat box and I will discuss the answers. Okay. Erythromycin belongs to dash class of antibiotics. Let's check the chat box now. Number A, number A, number A. So I have answer. I have received the answers which are saying it's number A. So erythromycin belongs to, very good, macrolide class of antibiotics. So these are the macrolides, the structure of the macrolide. This is the erythromycin. Okay, this is the structure of the erythromycin when there is a X equal to OH. Okay, and here is the X as you can see, and erythromycin when X is equal to OMe. Okay, and this in the structure we have a lactone functional group here. As you can see, there is a sugar which is a clarinose, and then we have the amino sugar which is the desoaminose. Okay. So macrolides are the bacteriostatic agents. I have discussed what is bacteriostatic and what is bactericidal in the few questions earlier. I have already discussed it. Okay. The best known example for these compounds is the erythromycin, a metabolite isolated in 1952 from the soil microorganism Streptomyces erythraeus found in the Philippines, and one of the safest antibiotics for clinical use. The structure here it is mentioned is a 14-member macrolide lactone ring with a sugar and an amino sugar attached. The sugar residues are important for the activity. Erythromycin acts by binding to the 50th subunit of the bacterial ribosome. This is important how it works, the mechanism of action. Okay, so erythromycin acts by binding to the 50th subunit of the bacterial ribosomes to inhibit the translocation, but other mechanisms of the action also appear likely because erythromycin and chloramphenicol bind to the same region of the ribosome. They should not be administered together as they will compete with each other and be less effective. Okay, erythromycin is unstable soluble to stomach acid but can be taken orally in the tablet form. The formulation of the tablet involves the coating of the designed to protect the tablet during the passage through the stomach, but which is soluble once it reaches to the intestine. Okay, the enterosoluble. The acid sensitivity of erythromycin is due to the presence of the ketone here, and the two alcohol groups which are said to for the acid catalyzed intermolecular formation of the ketal. One way preventing this is to protect the hydroxy group. For example, clarithromycin is the methoxy analog of erythromycin, which is more stable to the gastric juices and has improved oral absorption. Another method of increasing the acid stability is to increase the size of the macrolide to a 16 membrane ring. Azithromycin contains a 15-membered macrolide where the N-methyl group has been incorporated into the macrolide. It is one of the world's best-seller drugs. Erythromycin is a semi-synthetic derivative of erythromycin and reached to the European market in 2001. The clarinose sugar of the erythromycin has been replaced with the keto group, and the carbamate ring also be fused to the macrolide ring. The true hydroxyl group can cause the intermolecular keter formation in the erythromycin have been marked, one as the methoxy group and the other is a part of the carbamate ring. So these are the other example of the macrolide, the azithromycin and the terythromycin. Okay. So whenever you are reading a particular class of the antibacterial agent, the things which you need to remember is the structure. If you don't remember, you cannot memorize the structure like if the structure is like bigger, like big structure like erythromycin and so on, you don't need to memorize. You have to just remember what are the example and what is the mechanism of action. That much will be fine for to remember. Okay. Now I will go to the next problem. Problem number 16 says how oxalidinones works. Number A, it inhibits the protein synthesis by binding to the 30S ribosome. Number B, it inhibits the protein synthesis by binding to the 50S ribosome. Number C, it unwinds the DNA double helix. Number D, it inhibits the DNA guidage enzyme. How oxalidinone works? So oxalidinone are another class of antibacterial agent and you have to identify the correct mechanism of action of oxalidinone. Your options are: number A, it inhibits the protein synthesis by binding to the 30S ribosome. Number B, it inhibits the protein synthesis by binding to the 50S ribosome. Number C, it unwinds the DNA double helix. Number D, it inhibits the DNA guidage enzyme. A, B, C or D, let me know which of the following is the correct answer. I will wait for a minute, then I will check the chat box. Okay. How oxalidinone works? Let me check the chat box now. So I think I have received answers. I have received two answers. One is saying it is number B. Both are saying it is number B. So let me check. So yes, number B is the correct answer. Oxalidinone works by inhibiting the protein synthesis by binding to the 50S ribosome. So oxalidinones These are the oxalidinones, okay, linazolide and rabizolide. So these are the structure of the oxalidinone class of antibiotics. So the oxalidinones are the new class of the synthetic antibacterial agent discovered in the recent years. They inhibit the protein synthesis at a much earlier stage than the previous agents and consequently do not suffer the same resistance problem before protein synthesis can start. A 30S ribosome has to be formed by the combination of the 30S ribosome and a 50S ribosome. The oxidinone bind to the 50S ribosome and prevent this from happening. As a result, translation cannot even start. Other agents that inhibit the protein synthesis do so during the translation process itself. Linazolide was the first of this class of the compound to reach the market in 2000, and by 2010 it was the netting sales of the 716 million per year. X-ray crystallography structure have revealed that the structure binds to the ribosome, and that has allowed the development of analogs which bind more strongly. Rabizolid is the one-stop structure that binds 10,000 times strongly as a result of the extra binding interaction, and this is also another example of the extra extension strategies. It is currently undergoing the clinical trials. Oxalidinone have the broad spectrum activity and are active against the bacterial strain which are acquired resistant to the other bacterial agents acting against the synthesis. Linazolide has a good activity against the most clinically important gram-positive bacteria, including MRSA. It can also it can also be taken orally with 100% uptake from the gastrointestinal tract. Unfortunately, there is a high level of side effect related to the same. So it is used as it is a bacteriostatic agent. It is a greater risk of the bacterial resistance developing. Okay. So the correct answer was number B. Now I will go to the next problem. Problem number 17 says which of the following antibiotics works by targeting DNA guidage? Your options are number A sulfonamide, number B tetracycline, number C quinolone and number D glycopeptides. Which of the following antibiotics works by targeting the DNA guidage? Sulfonamide, tetracycline, quinolones and glycopeptides. Which of the following antibiotics works by targeting the DNA guidage? Sulfonamide, tetracycline, quinolone, glycopeptides. Which of the following antibiotics works by targeting DNA guidage? Let me check the chat box. Okay, I have received answer which says it is number B. Let me check if number B is the correct answer or not. So the antibacterial agent that works by targeting the DNA guidage is this number C, quinolones. Okay. So this is the complete chart which I have already mentioned earlier also. So DNA guidance, they works on the quinolines, RNA polymerase, rifampicin works on the RNA polymerase, 50S subunit of the ribosome, macrolides, clindamycin, linazolide, chloramphenicol and streptogramins, they work on it. Okay, tetracycline and aminoglycoside, they work on the 30S unit of the ribosome. Cell membrane, polymyxin, vancomycin and bactricin work on the cell wall, the beta, the outer wall. Okay, the beta-lactam, penicillin, cephalosporin, carbamazepine, monobactam, they also work on the cell wall, and the folate synthesis is inhibited by the sulfonamides and trimethoprim. Okay, the conversion of power to DHFA, it is inhibited by sulfonamide, and DHFA to THFA inhibited by the trimethoprim. All this I have already mentioned in earlier. So DNA guidase is an essential bacterial enzyme that catalyzes the ATP-dependent negative supercoiling of the double-stranded closed circular DNA. Guidase belongs to a class of the enzymes known as the topoisomerases that are involved in the control of the topological translation of the DNA. Topoisomerases are able to change the DNA topology to facilitate functions such as the DNA replication and transcription. Okay, and the quinolones are the bactericidal. They inhibit the bacterial DNA synthesis in several ways, causing the rapid cell death. This is the quinolone. This is the this portion that binds to the enzyme. This portion binds to the also enzyme. This is the DNA binding domain, and this is the stacking domain. Quinolones bind to the DNA guidase or topoisomerase 2 in a complex blocking the further DNA replication. Okay. So quinolone was the correct answer. Now problem number 18, it says which of the following is the general mechanism of action for vancomycin? Your options are number A, inhibition of the metabolic enzyme, number B, inhibition of the cell wall synthesis, number C, disruption of protein synthesis, number D, inhibition of the nucleic acid transcription and replication. Which of the following is the general mechanism of action for vancomycin? Inhibition of the metabolic enzyme, inhibition of the cell wall synthesis, disruption of the protein synthesis, and inhibition of the nucleic acid transcription and replication. A, B, C, or D? Let me know which of the following you think is the general mechanism of action of the vancomycin. Inhibition of the metabolic enzyme, inhibition of cell wall synthesis, inhibition of protein synthesis, inhibition of nucleic acid transcription and replication. Already I think I have mentioned it for a couple of times, so I will give you only 1 minute, then I will check the chat box, okay? Which of the following is the general mechanism of action for vancomycin? Let me check the chat box. Okay, no answer. Okay, it's fine. I will just show you the answer and let us discuss it a little bit. So the general mechanism of action of vancomycin is number B, inhibition of the cell wall synthesis. So this is the structure of the vancomycin. So yeah, vancomycin is a crucial drug of the last resort, inhibits the peptidoglycan synthesis by binding directly to the D-ALA end of the peptide, forms a cap over the end of the chain and blocks the cross linking. I have already mentioned it earlier also that how it behaves, okay. So it inhibits the peptidoglycan biosynthesis of the bacterial cell wall, blocks the transglycolysis and transpeptidase activity, prevents the transpeptidation linking, and stops the bacterial cell wall maturation. Okay, so this there is a vancomycin. It caps on the on this particular form. Okay, so this that is how the particular inhibition of biosynthesis is the mechanism of action of the vancomycin. Now I will go to the next problem. Problem number 19 says rifampicin binds to dash. Fill in the blanks. Your options are number A, RNA independent DNA polymerase, number B, RNA dependent DNA polymerase, number C, DNA independent RNA polymerase, number D, DNA dependent RNA polymerase. Rifampicin binds to which enzyme? Number A, independent DNA polymerase. Number B, RNA dependent DNA polymerase. Number C, DNA dependent RNA polymerase. And number D, number C is DNA independent RNA polymerase. And number D is the DNA dependent RNA polymerase. Rifampicin binds to which of the enzyme? RNA independent DNA polymerase, RNA dependent DNA polymerase, DNA independent RNA polymerase, or DNA dependent RNA polymerase. Let me know in the chat box A, B, C or D which will be the correct answer for problem number 19. Let me check the chat box. Okay, so number C and number B. I have got two answers. Let me check. Number B or number C, which of the following is the correct answer? So the correct answer will be, it is number D, DNA dependent RNA polymerase. Okay. So rifampicin is a semi-synthetic rifampicin made from the rifampicin B, an antibiotic which is isolated from the Streptomyces mediterranae. In 1957, it inhibits the gram-positive bacteria and works by binding the non-covalently to DNA-dependent RNA polymerase and inhibiting the start of the RNA synthesis. The DNA-dependent RNA polymerase in eukaryotic cell are unaffected because the drug binds to the peptide chain not present in the mammalian RNA polymerase. It is therefore highly selective. The flat naphthalene ring, and the several hydroxyl groups are essential for the activity, and the molecule exists as a zwitterion giving to the good solubility in both lipids and aqueous acid. Rifaximin is another semi-synthetic analog of this which approved in 2004 for the treatment of the diarrhea and E. coli infection. So this is the rifamycin B and rifamycin, and this is the rifamycin, and the enzyme which it binds is the DNA-dependent RNA polymerase. Okay. Now, last question of today's session. It says aminoglycosides work by blocking dash during the translation. Your options are number A, tRNA binding, number B, translocation, number C, peptide chain transfer, and number D, incoming amino acid. Aminoglycosides works by blocking dash during the translation. Number A, tRNA binding, number B, translocation, number C, peptide chain transfer, and number D, incoming amino acid. Aminoglycosides works by which? By blocking of the which of the following? During translation, where does this aminoglycosides work? It blocks tRNA binding, or it blocks translocation, or it blocks the peptide chain transfer, or it blocks the incoming amino acid. Let me check the chat box because we don't have much time left. Okay. Okay, number C. I have got the answer number C. Let me check if number C is the correct answer or not. So aminoglycosides work by blocking translocation during the translation process. Okay, it blocks the translation. So basically, this is the stages which antibacterial inhibit the translation process. So in the translation means the protein synthesis, we have the 50S and 30S ribosomes together. Oxalidones binds to the 50S ribosome, I have already mentioned. Then after the forming this translation complex, we have the growing protein chain, and amino acid is taken coming with the tRNA, and tetracycline blocks by tRNA binding. Then we have the chloramphenicol, which blocks the peptide chain transfer, and last we have the macrolides block the translocation, also the aminoglycosides which blocks the translocation. Okay, so translocation, it is blocked by the aminoglycosides, also macrolides, they block the translocation. So there are different in this chart we have different antibacterial agents which works on the translation, different steps of the translation, all are mentioned together. So aminoglycosides, they work by blocking the translocation during the translation process. Okay. So this was the last question of today's session. So today what we have understood, we have discussed about the antibacterial agent. I have tried to give you the different antibacterial agents in different questions to just summarize the whole thing. Also we have discussed about the solid phase synthesis and the combinatorial synthesis also. And antibacterial agents are very important topic of your course. So try to read all these things, and if you have any doubt, any query, you can ask me in today's class also, but we don't have much time. So I am sharing my email ID. So this is my email ID. If you have any doubt, any query regarding today's class or previous any class, feel free to reach me through the email. Also, you can post your queries in the discussion forum, okay, of the NPTEL Medicinal Chemistry. So today it was our week 10 live session, and Thank you very much for joining and thank you very much for answering the questions. Okay, most of the answers you have given are correct, so it is very good. And I will share it by I will be sharing this PPT or also the recording of the class in shortly. So I have already shown you where to find the material, so I think it won't be any problem. So thank you all. I will close the session for today. Let's meet on the next Friday for the next live session, which will be on the next Friday, and it will be on the week 11 live session. Okay. So thank you all. I am closing the session for today. Please let me know if you have any doubt, any query via this email ID. Okay. Thank you.